Hello, my name is Jaehyun Kim, undergraduate student of Bio and Brain Engineering Department. Today I will give you a brief review about the last special lecture conducted by Professor Min-young Kim. Today's presentation is composed of 4 parts: introduction, methods, results, and summary. Let's start from the introduction part. In the last online lecture, I explained the concepts of regenerative medicine, central nervous system, and cell therapy. Combining these 3 concepts, the stem cell treatment for brain disorders could be possible. The actual example of stem cell of treatment of brain disorder could be described as this diagram. Combining the treatment of umbilical cord blood, erythropoietin, and rehabilitation can achieve enhanced performance of cerebral palsy therapy. Before explaining about the application of stem cell treatment, I think I need to review about the 3 core concepts: cerebral palsy, umbilical cord blood, and erythropoietin. Cerebral palsy describes a group of disorders of the development of movement and posture causing activity limitation that are attributed to non-progressive disturbances that occurred in the developing fetal or infant brain. It's associated with physical disabilities in human development, such as cognition, perception, and language ability. Moreover, it often leads to lifelong functional deficits. UCB, the abbreviation of umbilical cord blood, is simply the blood extracted from umbilical cord which connects fetus and placenta. It has been reported that UCB contains stem cells with variable therapeutic potentials, such as hematopoietic and neurogenic stem cells. Also, it has anti-inflammatory, anti-apoptotic, and neuroprotective properties, which makes it as a potential candidate of biopharmaceuticals. EPO, the abbreviation of erythropoietin, is originally a hormone secreted from kidney that takes part in the production of red blood cells. However, it has neuroprotective and neurorepair properties as well. Since it shares same cellular signaling pathway for cell survival with UCB, it was expected that there will be some synergetic effects in treatment of EPO with UCB. From now on, I'll review about the methods used for the new treatment strategy of cerebral palsy. For experimental group, only 10 months to 10 years old of children who diagnosed as cerebral palsy are included to maximize the reliability of this experiment. Any features that thought to be hampering the interpretation of results according to the clinical judgment of the principal investigator, such as genetic disorder or epilepsy, were excluded from the experimental group to avoid critical adverse effects and enhance reliability. Next, this experiment obeyed the placebo-controlled, double-blind, and randomized trial protocol. Each patient is randomly assigned to these 3 groups: PUCB, EPO, and control group. For the group does not be treated by UCB or EPO, they are treated by placebo UCB and placebo EPO to avoid placebo effect. Also, all participants, including family members, observers, investigators, and employees, were blinded to the group assignment to achieve double-blind test. Both EPO and UCB are treated to the patients by intravenous injection. Unrelated allogenic UCB was used for this experiment, and cyclosporine was treated together with UCB for immunosuppression. Autologous blood and albumin was used for the placebo UCB, and EPO treated 2 and 12 hours before the UCB or placebo UCB injection. 0.5 cc of albumin in 150 milliliter of saline or orange juice was used for placebo EPO. As a traditional and general therapy for cerebral palsy, rehabilitation was also conducted for the patients in all of the 3 groups. Intensive rehabilitation program was performed during the first 1 month, which consists of twice of physical and occupational therapy per day. After 1 month, the patients were discharged, and post-discharge rehabilitation program was conducted at least 3 times per week for 6 months. From this slide, I will tell you about the result of the research methods that I explained until last slide. The results could be divided into three parts: functional assessment, DTI, and PET imaging. To achieve functional assessment, gross motor performance measure and Korean-Bailey scale second version and WFIM was selected for outcome measurement. Here we can see the result of GMPN and WFIM test. Through GMPM and WFIM, we can measure the improvement of gross motor ability, social cognition, and functional independence of the treated patients. We can see the PUCB group has the significantly higher scores on the GMPM and WFIM after 6 months of treatment. That means the patients treated by PUCB improved their motor ability and social cognition. Another result of functional assessment is presented in this slide. BSID-2 is a test to measure neurodevelopmental process which can evaluate the performance of mental and motor function of the subject. We can see the PUCB group also shows significantly higher scores on the motor scale and mental scale of BSID-2. That means the patients treated by PUCB improved both of their motor function and mental function. Next, the diffusion tensile imaging is used for measurement of fractional anisotropy of the brain of patients. It is a type of MRI which allows the mapping of the diffusion process of molecules, mainly water, in vivo. It has been widely applied to study and treat of neurological disorders because it can reveal abnormalities in white matter fiber structures. Here is the result of DTI, translated as digitized values of FA. FA represents the effects on white matter integration, which provides a route for information flow inside the brain. Therefore, it correlates with the brain connectivity and brain function. Through this graph, we can see the FA increased both in right and left side of the brain due to the treatment of PUCB. And it means the white matter integration and brain connectivity has increased by the treatment of PUCB. By DTI, the correlation between changes of GMPM and PFA in posterior internal capsule is also measured. Since the internal capsule is the collection of projection fibers from cerebral cortex, we can easily infer that the FA of PLIC might be related to the motor ability of the brain. The result proves that this hypothesis is correct. The FA at the posterior portion of right and left PLIC and anterior portion of left PLIC were measured with the changes of GMPM. The result shows that there is a clear correlation between GMPM and FA of internal capsule. It means the integration and connectivity of internal capsule is related with motor function of the brain. Next, the positron emission tomography was used for measurement of metabolic activity of the brain of patients. The FDG was selected as a biologically active molecule for PET to visualize metabolic activity by the regional glucose uptake. The increase of metabolic activity in basal ganglia and thalamus in PUCB group is shown in this picture. Since both of the basal ganglia and thalamus are related in motor ability of the brain, we can infer that patients treated by PUCB consume more energy to perform motor function. The decrease of metabolic activity is measured by PET as well. In this picture, we can see the decreased metabolism in occipital and limbic lobes in the PUCB group. Considering that EPO was administered to both the PUCB and EPO groups, the different findings between the two groups could have caused by UCB action that affected complex brain network. Today we have discussed about stem cell for treatment of brain disorders, and I'll briefly summarize the contents of today's presentation before finish this lecture. The stem cell source used for this experiment was UCB, potentiated with EPO. Rehabilitation program also performed for cerebral palsy, a major brain disorder in rehabilitation medicine. Elevation in both mental and motor function was observed from the patients treated by UCB therapy. Therefore, This novel treatment could be a new therapy for cerebral palsy and other brain disorders. Thank you for your attention to my presentation.